Hey everybody, Mr. Murphy here. I'm going to take a look at chapter 3.2, which is a big one, all about proteins. So there are many different types of proteins and therefore many different functions of proteins. But very important is to understand that proteins are the workhorse of the cell. They are the molecules that are doing things inside of living organisms. When we first learn about how the cell works and things like that, and we look at the nucleus and it holds the DNA, and we often simplify it and say the nucleus controls the cell, right? It's kind of wrong, and sorry to be a part of that misconception, but when you're first learning about the functions of organelles, that's a helpful way to sort of think about the nucleus and its importance. But the incorrect part is that the nucleus is just housing the genetic information, and that genetic information is just what I said. It's information. It's not actually capable of doing things directly, for the most part, in terms of DNA. RNAs can do things very similar to proteins, kind of as we alluded to in the earlier 3.1 section. But really, it's the protein structures that are formed from those instructions that are doing the things inside of the cell. So enzymes are a major part of that, but they're not the only example of proteins. We have various defensive proteins like antibodies, which is super cool. We have different hormonal and regulatory proteins. So these are like signaling molecules that can then stimulate change within the cell in response to environmental conditions or needs of the larger organism. Receptor proteins that are all about sort of the connection and interaction between different cells, so on the surface of cell membranes, storage proteins, all sorts of things peppered throughout the entire cell membrane. A whole ton of different types of proteins that facilitate the movement of molecules in and out of the cell. Lots of things involve proteins. It's the molecule that is doing stuff inside of the cell. So more there listed. So the monomer of proteins are amino acids, and amino acids have this basic shape. I think this is a terrible representation of it, but we have the amino group here on the left-hand side, or the amine group. We have a carbon in the middle, and then we have a carboxyl group on the other side. Now, we've talked about this in class. This representation shows that this hydrogen from the carboxyl side is kind of migrated over here to the amino group. That is not something that I'm going to directly quiz you on or anything like that, but just understand that chemistry is a little more chaotic than the simple rules we like to define for ourselves. So if you're thinking about an amino acid as being this NH2, and then a C with an R group, and then this COOH, the carboxyl group, that's totally fine. This is just sort of showing how in an aqueous solution inside of a biological organism, the hydrogen can sort of float a little bit, and that primes it for the reactions that will occur. That R group, the side chain, is what makes each amino acid unique, and there are 20 different amino acids that we find across life. A significant point is that whether it's a bacteria cell or a fungus or a plant or an animal, it's the same amino acids within each, like the exact same physical chemical structure of those amino acids that are the building blocks of those, which we'll circle back to sort of as a really significant indicator of the commonality of the ancestry of life on the planet. So the different amino acids that we have, you don't need to memorize them. I will never ask you for any specific details by name, with maybe one exception that I'll get to. But just understand that in general, we have positive and negative side chains, which can then cause attractions. We'll see in a little bit. We have polar side chains that are not charged, but they're polar, so they act charged because of the distribution of electrons. There are some special cases, and this is the one, cysteine is the one that I would, if I were asking you a question involving this, I would give you enough information that you wouldn't have to know cysteine alone. But cysteine is one that has this sulfhydryl group here and will be responsible for a very specific type of bond in the structure of proteins. And then the last ones are the nonpolar side chains, which again, nonpolar molecules will tend to stick together because of very weak nonpolar van der Waal-related interactions. Okay, so here is the special case of cysteine. When two cysteine amino acids are situated near each other, and that they don't actually have to be next to each other. I don't think they could be next to each other. to do this on like a chain of amino acids, but somewhere in a protein you've got two cysteine molecules, and they can fold over and form this sulfide. The sulfhydryl groups can form what's called a disulfide bridge, which is a covalent bond that forms between them. That's significant because, again, when you hear covalent, we should immediately think like strong bond. And much of the structure of proteins is related to hydrogen bonds, which in great enough number can be meaningful and is meaningful, but doesn't have that strength, can't withstand environmental condition change nearly as well as a covalent bond. So, if you were ever asked about like a amino acid that's particularly resistant to the effects of pH or temperature or whatever, if one of the options of possibly explaining this was that it contains a lot of cysteine amino acids, there would be a reasonable support for that to be an explanation for why it's a particularly hardy protein. So when we look at polypeptides, which are these growing chains of amino acids, that happens through peptide linkages, which, just like a glycosidic linkage or an ester bond or a phosphodiester bond, it's just a name for the dehydration synthesis which occurs between an amino group and a carboxyl group. And here we have, again, representing this as NH3 on one side and COO on the other side. Doesn't really matter whether you look at this as an OH and this is just one of two H's. But the point is that we take the ingredients of water and we pull that out. It leaves a carbon that wants a bond, a nitrogen that wants a bond, and that carbon and nitrogen will bond together. And that's what we call the peptide linkage or peptide bond. And we get water. It's dehydration synthesis, condensation reaction, which means that we are building monomers into a larger molecule. The primary structure, secondary structure, tertiary, and quaternary structures of proteins can be a kind of a lot when you first approach it. But I would argue that it's a relatively simple idea. We just have to wrap our head around it in the right way. This is not like a step-by-step ingredient list for how to make a protein. This is just explaining that there are 4 different sort of levels that are all simultaneously potentially contributing to the shape of a protein. Proteins, whether it's enzymes or a channel protein in a cell membrane or whatever, all rely on shape. The shape is what dictates how it can react, because the shape is the arrangement of the different side chains, the arrangement of different potential hydrogen bonding sites. It's the thing that dictates like how a protein can grab on to other molecules. We like to think of like chemistry as chemistry. It's this sort of nebulous thing that allows for reactions to happen. And what we're trying to do in this class is rethink that in terms of the fact that if you zoom in enough, these are 3-dimensional molecules that have very specific shapes that dictate how they can react. And so when we look at a protein, there are 4 different things simultaneously happening. Primary structure is just straight up which amino acids happen to be linked together in this chain. If I have a whole bunch of cysteine molecule or cysteine amino acids, or I have proline and isoleucine and tryptophan, whatever, the point is that the characteristic of that protein is going to be different if I have different amino acids linked together. I use the analogy of Lego bricks to talk about monomers and polymers all the time. And the idea here is if we had 20 different kinds of Lego bricks with unique shapes and, side connections and whatever, which Legos I stick together is going to create something that has different potential. If you're just randomly grabbing Lego pieces and stitching them together, you may end up with something wildly different shape than if you did that again and got 20 random different Lego bricks. So that's what primary structure is all about. It's just the sort of what are the actual components that we're using to build this and how are they situated next to each other. The secondary structure is what happens when you have these things in this chain and you've got these amine groups with a potential that hydrogen sticking off, and you've got the carboxyl group with the electronegative oxygen double bonded to it, and how those can begin to have hydrogen bonds existing between the amine groups and carboxyl groups of different amino acids. And so local amino acids may start to attract each other and form this sort of spiral so that the amine group on one amino acid is attracted to the carboxyl group of an amino acid just a couple down the chain. And so I can sort of form this twisting shape, and that's represented here. We also see interactions like in a larger sense that like entire branches of an amino acid could actually sort of fold over on itself and align these amine groups and carboxyl groups. And so the amine group here is the blue nitrogen with the white hydrogen, and then the carboxyl group is this red oxygen here, and that's just a positive polar region and a negative polar region, and they attract together. So hydrogen bonding, individually weak, but in great enough number, it contributes to a important shape aspect that we see within proteins. So that's the second sort of simultaneous chemical attraction that's going on. The third one, tertiary structure, is all about those side chains. So we have 20 different amino acids. Some are positive, negative, polar, nonpolar. Some are the special cases with sulfhydryl groups or whatever. This is where we really start to get folding and wrapping of these amino acid chains because of just hydrogen bond interactions between various different side chains, or ionic attractions between positive and negative regions, nonpolar regions sticking together. And, I describe it as just all happening on top of one another. Each individual amino acid is attracted in like 40 different ways. That's definitely an understatement. Like 4,000 different ways potentially to all these different amino acids, and there's going to be sort of predominant forces that lead to the final shape of the protein. But it's not just like this protein folds and then it's just folded. It's a constant struggle. These amino acids are constantly being attracted to one another in a variety of different ways, but at the end of the day, there is some sort of arrangement that wins out based upon what amino acids happen to be placed where, and how larger groups can sort of work together to all sort of meet their hydrogen bond attractions and things like that. Pepper in there some disulfide bonds, which stitch it together a little bit more permanently because of those covalent links. But that's what tertiary structure is. So we have sort of regions that show the secondary amino carboxyl attractions, the hydrogen bonds between those. We see the actual amino acids themselves in the chain. That's the primary structure. We have these regions of spirals, again, secondary. And then we have just like the big level folding, that's tertiary. Again, it's not step by step. It's not like here's primary, then secondary, then tertiary. It's just all happening at the same time. We just define it as like different levels of attraction. So tertiary structure is where all polypeptides end, and that's then the three-dimensional shape of a polypeptide. We'll circle back to quaternary structure in a second. So we talked about disulfide bonds. It's predominantly hydrogen bonds. There's hydrophobic and van der Waal interactions, very weak but meaningful, just to drive like what amino acids want to sort of be attracted to which other ones. The positive negative ones can form little salt bridges where they're an exchange of electrons, and therefore the positive negative attractions. All of that is happening all at the same time, and in the end you get this three-dimensional shape. So here's a good representation of that. This just looks like nonsense, I know, but it's representing the different pleated sheet, beta pleated sheet, which is the secondary structure, the alpha helix, which is again secondary structure, and then sort of nondescript amino acids, just all in a tangled, jumbled mess. It's subject to denaturing, and denaturing is just a change in shape. Denaturing does not mean complete destruction, necessarily. It doesn't mean complete loss of function either. A denatured enzyme, a denatured protein, may still do its job, just not as well. Since it's so dependent upon its shape, any changes or challenges to the shape of a protein is definitely going to hinder the ability of the protein to do its job, but does not necessarily destroy it. If it is sufficiently pushed, sort of with tons of heat, if you boil the peroxidase enzyme in our catalysis lab or anything like that, it's going to stop working because you're going to start breaking covalent bonds, and you can't undo that. But if you just heat something up and then you allow it to cool back down without breaking any of those more permanent bonds, it'll return to its original structure when its conditions are ideal and correct again, just based upon what those chemical interactions are. Eventually, the ones that are the predominant forces will win out, and the shape of that protein will come back. Then we want to return to the idea of quaternary structure. So denaturing is occurring to polypeptides, and polypeptides are the primary, secondary, tertiary structure of proteins. A polypeptide chain is a single amino acid chain that's done all the folding. Quaternary is kind of a special case where you have multiple polypeptide chains that bond together through hydrophobic, ionic, and hydrogen bond interactions. So this is where you have like different polypeptide chains that may come together to form an even larger protein. That's the idea of quaternary structure. Some proteins are just a single polypeptide chain. Others are larger or more complex or are doing multiple things or whatever, and so it's multiple polypeptide chains sort of conjoined together. And here you can see this is an example of hemoglobin, which is just a go-to example. It's a very common protein in a whole host of organisms, as it is related to the capturing of oxygen, and is made out of 4 different polypeptide chains. So each of these 4 different colors here are 4 different polypeptide chains that have been constructed with the various amino acid interactions, and then they came together on top of that with even more hydrogen bonds, ionic bonds, and all of that. So now if we look at either a single polypeptide chain or a quaternary, multiple polypeptide chains together, there are 4 fundamental things which influence the shape of proteins. We sometimes think of these as the 4 things that affect enzymes. I want you to lock it in more broadly. Proteins in general are all affected by these things. And we've talked about it in class, but just to go through it again, temperature, concentration of pH, or concentration of H plus ions, which is pH, nearby polar substances or nearby non-polar substances are all going to challenge that sort of stability of where that protein has landed. Remember, proteins have so many different interactions all on top of each other, but it eventually settles into a mostly consistent shape. And you notice I don't stop moving my hands because it's never just this static, like locked in thing. It's always kind of pushing and pulling, but it's got the general shape that it needs. As soon as we introduce either more energy and they start moving around more, you start losing that shape more. That's temperature. If you introduce more H plus ions or OH minus ions, that's acids or bases, maybe those protein chains start being attracted to those positives or negatives. It starts losing its shape, denaturing. If you put lots of polar substances, a bunch of salt molecules, Na plus, Cl minus, that can start pulling on the different amino acid side chains and changing the shape. Or non-polar substances can attract the other non-polar amino acids. The point is we're just disrupting the homeostasis of where did this protein settle? And now if we change the environment, we change the rules of what are the dominant chemical interactions that are forming the shape of this thing. And all of these need to be carefully managed inside of our bodies. Otherwise our proteins stop doing their jobs. And since proteins are the workhorse of the cell, that could be pretty bad pretty quickly. So, we maintain a 98.6. We maintain a pH very close to 7. We have a maintenance of polar substances inside and outside of our cell through various channel proteins and things like that. Our body is constantly maintaining these things in order to preserve the intended ideal conditions for our proteins. Which brings me to one last point on this, which is a given organism is going to have a particular unique set of ideal conditions, which is why if you look at certain proteins, even if they're proteins that are prevalent across many different kingdoms of life, you're going to see that a tree, which may use a particular enzyme or something that's also found in us, may have a different build or structure to that protein depending upon what its ideal conditions are. So that is the end of this intro into proteins. We'll dig more deeply into it with enzymes in the next section. If you have questions, let me know.